Okay. All right, so we're going to start with reviewing for the final exam. I will, of course, finish the review on Wednesday. What I'm going to start reviewing, though, is the Chapter 30 stuff, more specific problems and concepts than the topics that we talked about last week. So, I mentioned last week you could potentially get a question where you would need to use the Wobble table, the Wobble base pairing table, in order to answer particular questions. One style of question that is very common is, here are all the codons of serine. Here are all 6 codons of serine. What is the minimum number of tRNAs you would need to match to all 6 codons? Now, we know the maximum would be 6, because that would be a 1-to-1 match for all 6 codons. But the minimum one, we have to figure out a couple things. The reason why you can have less than 6 is because of the Wobble base pairing concept. And if you guys don't remember what Wobble base pairing is, basically, let me draw, I guess, A, C, G. Here's the mRNA. And tRNA. The Wobble base pairing phenomena, or concept, is the fact that the third base in the anticodon can undergo sometimes non-canonical base pairing, or the first base in the anticodon matching to the third base in the codon. So this is the codon down here. This is the anticodon up here. And this spot, this base pairing, can sometimes experience non-canonical base pairing. What I mean by that is, well, normally G binds to what? C. So C can definitely be at that spot. But according to our Wobble table, sometimes, for whatever reason that we don't care about, sometimes G in a codon combined with U in an anticodon. So U can also go there. All right, and I think that's the 2 options. So how are you supposed to read a Wobble table? Well, look at your codon. Codon has a G there. Look in the table for everywhere that there's a G in the codon spot. That's right here and right there. A G has the potential to match with C, which we know that's normal. But also, on the Wobble table, they say that a G can sometimes match with a U. So that's why over here on the anticodon, I've written down C and U here. This indicates Wobble base pairing that this G has potential to bind to multiple things. So, because of this concept, because of this Wobble weird base pairing concept, we can try to figure out the minimum number of tRNAs we would need to bind to everything there. One thing to watch out for is the first 2 bases in a codon must undergo canonical base pairing, right? So, in other words, when I look at my serine codons, I know there are 2 groups to worry about, which I'm going to draw a line right between them right now. There are 2 groups of codons and serines to worry about. They will be different. They must have different tRNAs, because when we look at the first 2 bases between them, they're different from each other. So on tRNA, that's the canonical base pairing spot. So let's start with this group first. Let's figure out how many tRNAs would we need to take care of both of those codons. Well, I'm going to move this down here so we have some room. In an anticodon, we know because these first 2 bases are canonical that in the anticodon, I'm going to draw from 3' to 5', because everything up here is 5' to 3'. What must go on this end of the anticodon? A U, right? Because that's canonically base pairing the A's in the codon. Remember, the first 2 bases have to be canonical base pairing, no exceptions. The next base in the anticodon has to be what? C, because the second base in the codon is G, and therefore on the anticodon, it must be C, because there's no exceptions. The first 2 bases in the codon must experience canonical base pairing. What about the third one? I know there's 2 bases up here, but is there a potential anticodon base I can use that would take care of both of them? G, let's see where that is. Well, here's C and U in the codon table. Notice they both can be taken care of by G in the anticodon side of this table. So, if I just happen to have an anticodon that looks like this, a tRNA that looks like that, I've already eliminated 2 of my codons. So far I've reduced the number of anticodons that I would need from 6, and now we've eliminated a couple options here. So we now have to take care of the rest of them. Let's try that now. All right. So far I've taken care of these 2 with the minimum number I could take care of. Now I have to take care of the rest of the 4, and let's see what's the minimum number of tRNAs I would need to take care of the rest of the 4. All right, so I'm going to redraw these down here so we have room. Okay. The first 2 bases in the codon must experience canonical base pairing. So I know that in my anticodon, this spot has to be A and this spot has to be G, right? Because they all start with UCs. So that must be AG, right? Now for the slightly complicated part. What is the minimum number of anticodons I would need to take care of these 4 right here? 2, exactly. And in fact, there's 1 anticodon that would take care of 3 of those codons. What base should I put here to take care of 3 codons? I. Let's see. I'm going to put down an I here. Let's go look back up here. Notice that I in the anticodon spot can take care of A's, C's, and U's. So if I put an I here, it would take care of that, it would take care of that, and it would take care of that. Just from 1 anticodon, it has the ability to bind to 3 different codons. All right? Well, we're left with the last one, so we can honestly make up whatever we want for this one. A, G, and I guess we could put a C here. It doesn't matter. We need 1 more anticodon to take care of this last codon, so that would be that one. So this 1 anticodon took care of these 2. This 1 anticodon took care of 3 different codons, and this one took care of the last one, which we couldn't figure anything out for. So the minimum number of anticodons we needed to take care of all 6 codons of serine was what? 3. That would be the answer to the minimum number of tRNAs you would need to take care of these codons. Yes?
Would you also be able to use AGU instead of AGC?
For this one, yeah, you would. But the minimum number, so yeah, AGU would work here, but the minimum number would be either one of those. Yeah. Question?
So for that first one you did, you couldn't use I instead, so that you could only use minimum of 2?
Yeah, so for this one, what happens if we were to use I? Then this one would be UCI, would still work, but remember that this looks different from that because the first 2 bases are different. Yeah, that's why I split it up into 2 groups, because the first, it would still be a minimum of 3, yeah. Okay, you can do this for any amino acid in that table, so very likely on the exam you'll be given a different amino acid other than serine. I think on the worksheets I give you leucine for practice if you guys want to try that. But that's the procedure of how to solve a question that's like that. All right? Any questions on that one? Yes?
For the first one you did, ACG, was it supposed to be AGC or?
Which one? This one up here?
Yeah, you did the coding and
AGU and AGC, you need the U to match with A, and you need the C's to match with G's. Right? Yeah, and then the third base can be whatever the Wobble table says it needs to be. Okay. The first 2 bases, though, have to experience canonical base pairing. All right. So try this for whatever amino acid that you want. A lot of them can be reduced to less number of tRNAs than you'd expect. All right. Okay. Other questions on this style of question? Okay.
I have a question.
Yes.
I had to learn that engine I
Yeah.
creates the whole sequence process, right? So it can only be used at that beginning time, not down the.
Oh, you're talking about the I? No, this one can this can be a tRNA anywhere. You might be thinking of the AUG thing, the start codon. Yeah, that's its own thing. That's on mRNA. AUG is the start codon in every organism. The tRNA, I don't know off the top of my head what the tRNA would look like, but there's only one codon for methionine, so there would, at minimum, be only one anticodon for methionine. Yeah. All right. So let's try another complicated sounding question. Let's see. Some of these I did go over last week, though, so you might know some of these concepts already. If the coding units in the genetic code, if instead we had a triplet code, we had a unicode, how many possible combinations could I get out of a unicode? 4. Here's what this means. So remember the fact that we have a triplet code. What that means is we have 4 possible bases in mRNA. 4 bases in mRNA. And if we take 3 bases at a time, 4 to the power 3, we can create 64 possible combinations. Those 64 possible combinations will code for your 20 amino acids and 3 stop codons. So that's with the triplet code. For a unicode, if some unknown brand new organism was a unicode, one code, well, there's still 4 bases, but this time you only take 1 base at a time. The maximum number of combinations you can create is 4. All right? So at best, it can only code for 4 amino acids. It's very unlikely any organism anywhere goes by that. By the way, again, every organism on Earth, no matter what domain it's from, is the triplet code. This is all theoretical when I'm giving you Unicode and let's do a doublet code. What's the maximum number of combinations we would get if it was a doublet code? 16, right? Right? 4 bases, doublet code, we only get 16 possible combinations. Now you actually can see why we don't have a doublet code, because if we had a doublet code, we could only code for 16 amino acids, not 20. This is why we don't have a doublet code. So you can do this for any number that you want, and of course, since it's the final exam, you better bring a calculator, because there will definitely be some questions from exam 1 that you'll need a calculator. And for this style of question, if you were asked to calculate 4 to the power 4, you can figure that out too. All right, I don't remember what that is. What is that? 4 to the power 4, 64 times 4? I don't know, 256? 128? Who knows? Not my problem. But yeah, so when they ask about maximum number of combinations you get for Unicode, triplet code, doublet code, whatever code, is just 4 to the power whatever. All right, easy. Another way they could ask this is, what's the minimum length of code you would need to code for an organism that requires 100 different amino acids? Would a triplet code be enough to code for 100 different amino acids? The answer is no, because there's only 64 combinations. So the minimum length you would need, therefore, is 4, a quartet code or whatever. All right. So there's several different ways they could ask it based off this simple math. So just keep your mind open for those types of things. Any questions on that before I move on to the next concept? All right. So, oh yeah, a reminder on all those different vocab terms we associate with the code, with the genetic code, right? I know we went over it last week, so I'll only briefly talk about it. Universal, That vocab word basically means the genetic code is the same code in every organism. So a particular codon in a prokaryote will code for the same amino acid in both the prokaryote and the eukaryote. It's a universal code. So all organisms follow it. Degeneracy, a degenerate code. This basically means one amino acid can be coded by multiple codons. One amino acid can be coded by multiple codons, yes?
For universal, there was something I think in the PowerPoint that said like it's only for the genome, and that's why the mitochondria difference because the mitochondria has like a different genome or something.
The universal part is the fact that all those codons, no matter where, will code for the same amino acid. Unless you're talking about that, literally the only exception, which is tryptophan, can sometimes be coded by UGA. It's one of those weird quirks that you had to memorize on that one slide. But yeah, the phrase universal is more general. It's like for example, if I pull out a codon table real quick. If you have UUU, doesn't matter what organism it is, it'll always code for phenylalanine. That's what universal means. Okay, so universal, degenerate, non-overlapping is another one. Non-overlapping. Non-overlapping genetic code basically means the following. Is that we read the genetic code 3 bases at a time without repeating any reading. So what I mean by that is, here's an example of non-overlapping, which is how it actually works. And here's an example of overlapping code, which is the incorrect way. Here's an example of overlapping, which is definitely wrong, is that you're rereading bases a little bit at a time. That's overlapping. We don't do that. All right? So all these different vocab words are in your PowerPoint notes. Definitely review it. There's a couple more, but I'm just reminding you that that's probably a very important topic, and I did go over it last week, so you can look at the video there too. All right. Okay. Prokaryotic translation is split up into 3 steps, right? Initiation, elongation, and translation is what I'm saying, not transcription. I'm talking about translation, which is going from mRNA to protein. And there's initiation step, elongation step, and termination step. And there is an animation in the PowerPoint notes I highly recommend you look at and that you use, because it goes over all the different concepts and components. And you know what? Because we had proms last week, just going to briefly check if I'm able to do it today on the computer. If not, good luck. Let me just check it real quick. It's in the PowerPoints. The link for it, it's in the PowerPoints, yeah. So I'll load that for a second. Well, let me just draw a couple things while that's going. Of course, today it's slow because, you know. I guess it just never wants me to go over it with you guys. All right. I'll check back again in 5 minutes. Maybe it'll be there. All right. There's animation, and I don't know, did Dr. Aces get to that yet, the animation yet in class?
Yeah, I did it.
All right. So that animation is the one that I'm talking about. Make sure you look at it. The link is in the PowerPoints for 30B. Important concepts from that. What are the components you need to perform initiation in prokaryotic translation? What are the materials that you need? Well, obviously you need the small ribosomal subunit. Some of these are pretty intuitive. It makes sense. You need the small ribosomal subunit. You need the mRNA as well. Right? Because translation, you're going from mRNA eventually into protein, so you need the mRNA. It turns out, I wish the animation was working, is that you would see the small ribosomal subunit binding to the mRNA. Other stuff you need, though. In initiation, there's a category of proteins that help everything initiate. They are called initiation factors. Easy. All right. I believe there's 3 of them. You do have to know that there's 3 of them in prokaryotes. You don't have to know what 1 does versus 2 versus 3, just that there are 3 of them. What's the energy unit that we need to power this whole thing? GTP, you definitely need that. Let's see, small mRNA, initiation factors, GTP. Oh yeah, one more thing. The very first tRNA. What is it holding on to when we talk about prokaryotic translation initiation? It's a tRNA holding on to what amino acid? It is methionine with a modification on it called a formal group. That will be written as, it is methionine, just a slight modification to it. Always the very first amino acid in prokaryotes will be a formal methionine. All other methionines that show up will still be regular methionines, and they'll still be coded by AUG, like all the other methionines. It's just this first one will be a special one. Yes?
Do you need the large ribosomal subunit too?
The signal to the very end of initiation, and that's the last part on here. So everything else has to come first. The very end is the large ribosomal subunit. In fact, when this thing shows up, and you do need all of these, that's what potentially you may be asked about, but mechanically, if you want to visualize what happens, the only way for the large ribosomal subunit to come in is if the initiation factors leave and GTP is hydrolyzed into GDP to jumpstart everything. Imagine that GTP is hydrolyzed into GDP to jumpstart everything, and we no longer need the initiation factors, so those leave just when the large ribosomal subunit is coming in and fusing with the small ribosomal subunit. So these are the components you need to initiate prokaryotic translation. All right? Questions on that part? Okay. I guess he hasn't gotten to the eukaryotic translation stuff yet, right? So I'll talk about some components of eukaryotic translation. Actually, let me pull up the slide that I'm talking about. Nope. Did he get to eukaryotic translation yet? Or is that what he's doing today, or is he reviewing today? Anyone have any idea?
I honestly think he's reviewing.
He's just reviewing?
I don't know.
Because sometimes he doesn't include eukaryotic translation, so I do not know yet if he has decided. That's why I'm asking if he's covered that in class yet. All right, you guys might be lucky. You guys might not need it. I do not know yet, so I will at least very briefly talk about it then, for you guys' benefit. He may, because it's very brief. There may be lots of slides on it, but he usually doesn't care about most of it. It's like 1 or 2 slides that I'll bring up right now. I believe The majority of differences are on these two slides here. These numbered slides at the end of 30B, where you have these number 1, 2, 3, whatever. So, differences between prokaryotic and eukaryotic translation. So you notice a pattern in this class as we talk about prokaryotes in detail, we bring up eukaryotes a little bit for comparison's sake. So here's a list of differences between prokaryotic and eukaryotic translation. 1, we've known this for a while. Eukaryotic ribosomes are larger than prokaryotic ribosomes. Pretty easy. This one is interesting. Eukaryotic proteins begin with regular methionine, not formal methionine. So remember, in prokaryotes, the very first amino acid is formal methionine, but eukaryotes, it's regular methionine. Instead, what eukaryotes do to figure out the first amino acid is they have a special tRNA for it. So I believe that's listed here. Yeah, they have a specific tRNA for it in eukaryotes. It's a modified tRNA. Think of it as an initiation tRNA in eukaryotes. So contrast that. I'll repeat again. Prokaryotic proteins begin with formal methionine and regular tRNA. Eukaryotic proteins begin with regular methionine, but it was brought to the ribosome with a special tRNA, with an initiator tRNA. So that's eukaryotes. Oops. Remember the phrase polycistronic versus monocistronic. They're reiterating that here in blue. Polycistronic, again, is one mRNA, multiple proteins, multiple genes would be encoded in a single mRNA. But in eukaryotes, one mRNA, one product. This one's interesting. In prokaryotes, we learned there were 3 initiation factors for translation. In eukaryotes, there's some arbitrary number. I think they even list it later. It's like 17 or something. Just know there are more initiation factors required in eukaryotes than there are in prokaryotes. This one is quite important, and sometimes it goes over it in detail. Let me see where the other slide is for this. Oh, maybe he doesn't care this semester. I'll go over it anyway, but it may not matter for the final. I'll find out by Wednesday. Is this fact here? Translation in eukaryotes is regulated by phosphorylation of one of those initiation factors. The shorthand notation of that, if you see this lowercase e with IF, they're talking about eukaryotic initiation factor. That's what the lowercase e means, is eukaryotic initiation factor. The translation process is regulated by either the phosphorylation or the dephosphorylation of this initiation factor. And that's listed on here. And I believe the last 2 he doesn't usually ask about. Yeah. So the first 4 are the most important components. Yeah, diphtheria, he doesn't care about diphtheria. Yeah. So the first 4 components are the most important components when you're contrasting eukaryotic translation with prokaryotic translation. So if he has time to cover that, if he hasn't yet, those are the 4 main things I've ever seen him ask about when he talks about eukaryotic translation on the exam. All right? So I will move on from that. Oh, this is a good one. Okay. Let me see. Oh, just in case. Very important. If you're given a codon table that looks like this, don't panic. It's different. It looks different from the one in your notes, but here's how you read it. This is the first base in the codon. That's the second base, and that's the third base in the codon. Okay? So, as an example, ACU codes for threonine. All right? So just in case this version shows up on the exam of the codon table, that's how you read it. First base, second base, third base. All right? Yes. So if you get this version of the codon table, the first thing you may notice, it looks different in terms of structure compared to the one you're given in your notes. It's actually done so that it's a little bit easier to read. These bases over here represent your first base. So if you're given a codon ACU, how do you find it in this table? Well, the first base is A, so I'm going to look in this region. The second base is C, so I'm going to look there. So I align those two together. And the third base is U, which is that one. So aligning those three components together tells me that ACU codes for threonine. Oops. Codes for threonine. All right. So this one you can Google codon table, and this is like one of the first Google Images ones on there. You may also see this at the end of one of the practice exams as well. But this may be the table that we end up using. What was I going to say? Oh, okay. So, very quick, and then we'll end. I want a good example here. Let's say there's a mutation. There's a mutation from the codon CGU to GGU. What happened? What type of mutation do you have here? There were two types of mutations that you're going to need to know. And just realized maybe I forgot to talk about this. I'll talk about it during the final exam because there might not be enough time to talk about it now. There's two types of mutations, general mutations at the protein level. Missense mutations and nonsense mutations. You may have heard this in a genetics class. A missense mutation, very briefly, is a mutation that causes you to code one amino acid, and now the mutation causes you to code a different amino acid. So whenever you have a mutation that the end result is you have a different amino acid being coded, that's a missense mutation. So I'm going to put change amino acid here. The other type of mutation, nonsense mutation. There are others, but these are the two main ones we care about in this course. A nonsense mutation is a mutation where instead of coding for an amino acid, you've now switched it to a stop codon. All right, so if before you used to code for a regular amino acid, you have a codon, you go look in the table, it's supposed to be a regular amino acid, but the mutation ends up switching it to a stop codon, of which there's three options for you. That is a nonsense mutation. The way you can quickly identify a nonsense mutation, and I will go over this at the final exam since we're running out of time, short explanation. The quickest way to identify a nonsense mutation in the lab, and conceptually speaking, is your protein got a lot shorter than normal, right? If you end up having a premature stop codon, what that means is that translation is halted prematurely. Therefore, the protein that is normally made is now a lot shorter than it's supposed to be. That's the quickest way to identify a nonsense mutation. The quickest way to identify missense mutation is a change in amino acid. And I'll go over more in the final exam review.